So the first question, basically, again, I wanted to drop down from a 30,000 foot and then maybe tackle it a little bit more seriously, is what is, from what you've seen and from your extensive background in vaccinology, why do you think people are just in general having such an issue with getting this vaccine or having the fears about getting the vaccine in general, this particular vaccine? So let's say Moderna's vaccine.
So I think it's a few things. I think one is that people, for example, are more frightened of an anthrax vaccine or of an Ebola vaccine than they would say be for what is a more typical pediatric childhood vaccine, because the disease sounds scarier. So that if the disease sounds scarier, if the name is scarier, then they believe that the vaccine would be more dangerous. So that's number one. Number two is that it is a new technology. I mean, it's this messenger RNA technology is something with which we have no commercial experience. And the nature of the technology is it's a genetic technology. I mean, you're not being inoculated with a live attenuated virus or a whole killed virus or a viral protein. You're being inoculated with a gene that codes for that viral protein. Your body makes the protein. Your body makes antibodies to the protein, and that's a little scary. I mean, that's probably the most common question I get asked is, can it alter my genes? So I think, and that in combination also with the fact that it does have a difficult safety profile. I mean, if I've just gotten my second dose of the vaccine, and you can experience headache and fatigue and fever and muscle ache and joint ache and back pain, et cetera. So it lasts for a day or two. It does knock you out a little bit, this vaccine. So I think all those things make it a little scary and make people resistant.
So on something, I think it's interesting that somebody would ask, and this is maybe somebody that has a little bit more understanding of genetics, but not quite, that to say our level, is, you get a question about mRNA actually altering somebody's genes. How do you, where's the level of response for that? I mean, you could go, I imagine you could go, well, it's RNA, not DNA, and your chromosomes are made up of DNA. Where do you go with an explanation to the general public? How in depth do you go to sort of, one, explain the answer, give them an actual valid answer, but two, something that they can understand?
Well, I try and give them the scientific explanation and try and make it understandable. So for example, first of all, you're inoculated with this messenger RNA, which then enters your cells to then be there with all the other messenger RNA that's there. I mean, we all have messenger RNA, thousands and thousands of copies of messenger RNA in our cells, in our cytoplasm. So your cells have the nucleus, and outside of the nucleus is the cytoplasm. This particular messenger RNA just stays in the cytoplasm. It doesn't cross the nuclear membrane and go into the nucleus, which is where DNA resides. In order for messenger RNA to go into the nucleus, it actually requires something called a nuclear access signal, which it doesn't have. So therefore, it can't enter the nucleus. Even if it entered the nucleus, which it can't, it's RNA, it's not DNA. So it can't insert itself into DNA because it's a different molecule. It's two different languages. One is Spanish, one is English. They're two different languages. So it would have to be translated to DNA. The enzyme that translates RNA back to DNA is something called reverse transcriptase. That's an enzyme. That enzyme would have to be there, which it's not. I mean, the vaccine doesn't contain that enzyme that sort of translates RNA back to DNA. Even if it did, even if it contained that enzyme, which it doesn't, and it became RNA, became DNA, it still has to insert itself into DNA. In order to do that, it requires another enzyme that allows it to integrate into DNA, which is called, not surprisingly, integrase, which it also doesn't have. So there are three solid reasons for why it is not just a small chance that RNA can't affect DNA. There's a 0% chance. I mean, you have a better chance of getting this vaccine and developing X-ray vision than you have of this in any way negatively affecting your DNA.
So for someone like me that teaches virology and advanced virology and cell biology, I can understand all three of your answers and right down to the level of integrase and such. But I'm, the average person that's walking around out there. Are you going to still, give me that sort of level of explanation, or are you going to go right to, you have a better chance of getting X-ray vision than this altering your genes? That's where I'm stuck. I go in and I start giving that level of information to people, and I just see, eyes start rolling, or and such. Where's the sweet spot when you're talking to people that aren't, again, people that understand, you know, the ins and outs of RNA, DNA transcription, translation, what a reverse transcriptase even is, and such.
I still give the scientific explanation. I mean, I try and make it understandable. You know, you try and cross a wall that you can't scale. You try and you know, it's two different languages, and so you have to be translated back to one. It doesn't have the thing that allows it to do that. Can't integrate because it doesn't have this enzyme. That's what I give. I give the basic science information and give people credit to believe that although they may not understand the science exactly, it does sound right.
Okay. And do you typically get a reasonable response with something like, oh, okay, I mean, that makes sense, or are you brushed? I mean, how does that work? And again, I'm talking with, you know, your average sort of everyday person that might not be a scientist.
I think people generally buy that, and then you make a little bit of joke at the end, and then, you know,
then it works out okay in that respect. How about healthcare workers? I mean, do you find yourself having to give some sort of explanation to healthcare workers separately versus, say, you know, a nurse? Well, actually, a physician or a nurse, I would hope, would have a better understanding. I know because I teach in the science and also in the nursing department. They have a little bit, a nursing major or a nurse, an RN, has a little bit more, at least a little bit more understanding of what a virus is, what a vaccine actually does. You know, a basic understanding of what antibodies are, T cells versus B cells and such. Does your level of explanation change there, or do you just pretty much leave it at the same level?
Yeah, it's at the same level, roughly 8th grade level. I mean, kind of newspaper level is the way we explain. We created a vaccine education center 20 years ago, and we made the decision to try and explain things at roughly the 8th grade level. So now the CDC with their vaccine information sheets will largely go at the 5th grade level, which whereby I think you lose a lot of sophistication. So we chose not to do that. And nor do we choose a sort of the 11th grade level, which is Scientific American. We go for the newspaper level explanation. And we don't distinguish healthcare workers from, including doctors, from the general public. We think they all roughly have the same level of understanding of these things.
That's great. And have you seen actual physicians? Have you seen doctors say, I'm not getting the vaccine. I'm too unsure about it?
Yes, that's right.
Really?
Yes, doctors and nurses. Yep.
Really, that absolutely blows my mind. And is there a rationale? Is there a common rationale behind that, or is it multivariant? It's that, you know, well, I don't like the side effect idea, so on and so forth.
It's kind of a free-floating angst. You know, it is hard to watch yourself being injected with a biological, especially a biological you might not understand very well.
I see.
So I understand the fear. I just think that if you it's okay to fear this. It's okay to be concerned. And so you find out what people's concerns are and try and explain that. And people should be amenable to that reason. Some groups, so for example, there are African-American nurses in our hospital who are refusing this vaccine. And there it's not so much, you know, sort of a fear of this as a biological, it's a fear that that the powers that be aren't taking them into consideration, that they have been treated badly, ignored, made guinea pigs of, and they just don't trust the system. And you know, you're the system, so they're much less convincible.
And does that go back to, I mean, to be fair, does that go back to the ways that people of color and marginalized communities have been treated in the past by the healthcare system? I mean, is that an effect of that? I mean, do you have to go, you know, all the way back to the 1930s and such, or is that's, I mean, I would assume that's still happening today. They're still feeling that on a regular basis, right?
No, I think we've earned their distrust.
Yes.
And so we have to find a way to get them to trust us again. I mean, I was at, I don't know if you see, but I had this sort of scar over my right eye. That happened a few weeks ago, and from a fall, and I was in the hospital here, and the phlebotomist came by. I was waiting to get stitched up, and the phlebotomist came by, as an African-American phlebotomist, and I was watching the nurses go one by one to go get their vaccines, and I asked him whether he was going to get it. He was, you know, an African-American guy, and he said he wasn't going to get it. I said, so why not? He said, because I don't want to get the ones that they give to Black people. So we basically thought there were two different vaccines, one that was given to the white nurses and one that would be given to him.
Wow. Had you ever heard that before? Was that something new?
That was a new one.
Wow.
I mean, I told him, you know, I'm on the FDA's vaccine advisory committee.
Yeah.
There aren't two separate vaccines.
I can promise you. Yeah.
He wouldn't believe it.
Wow. That is amazing. But again, I mean, I think that's testament to how you've been treated and how your ancestors have been treated overall by the healthcare community. Absolutely.
Right.
Yeah, definitely. As far as your explanation of the vaccine, I mean, it sounds like you pretty much go into depth. Do you ever, as far as what mRNA is, how it's delivered, I've seen some of the recent speeches you've given talking about nanoparticle technology and such. Do you ever talk to people that maybe are concerned about the vaccine and, talk about, well, okay, this is what this technology is actually doing as far as, normally it would be B cell stimulation, but now we're having T cells. Like, do you sort of, like, one thing that I've done is, I was asked yesterday when I was actually getting tested so I could be on campus and teach it, and somebody was saying, well, what vaccine am I getting? Well, the one that's available, but what's this deal? I got, and this is a professor, I got, well, what's this deal with mRNA? And I'm sort of trying to explain, what B cells are, what antibodies are, and T cells, and so on and so forth, and why this is a different vaccine, and maybe why some of the side effects are different. Is that a level of detail that you sort of go into, explaining the immunology of how this works and how it could potentially be a better vaccine, so to speak?
Not really. I just try and get them to understand what a genetic vaccine is. In other words, unlike, say, if we were to make the SARS-CoV-2 vaccine in the more traditional way, which is to say, we would take just a purified protein, that SARS-CoV-2 spike protein, like we do for the hepatitis B vaccine or the human papillomavirus vaccine, and give the person the protein, or give them the whole killed virus, like with the polio vaccine, which is another way to give them the protein, or we use a live weakened virus, which is another way to give them the protein. What we're doing here is we're giving the gene to make the protein, and that's the critical difference. And I try and explain just the power of this technology. I mean, it is, when you look at the phase 1 trials and now the results of the phase 3 studies, it is possible this vaccination is better than natural infection. I mean, when they did the phase 1 trials, you compared it to sort of the range of neutralizing titers you were getting after natural infection, it was at the higher quartile of that range.
Right.
So, and that's why people, in large part, who've been naturally infected are still being asked to be vaccinated, because it will likely benefit you.
Right, and that's something, again, with going into the whole immunological aspect of it and talking about both B and T cell stimulation. That's where I try and land people. I don't know if that ever actually has worked in the past, but it's trying to say, this is much better, and it sort of lands you at. I've actually heard people say, and I know this is true for some vaccines and not true for some vaccines. Well, if I get infected, my natural immunity will be better, and I'm young, and so I'm going to survive it, and I'm going to be just fine. And in the end, I think this may be one of the vaccines, based on the information you just said and other information that I've read about, that's not true. This is one of those vaccines where it's actually better to get vaccinated than the actual infection as far as lifelong immunity goes. That usually, I mean, is that an area you tackle as well? Because usually for me, I got to tell you, it falls on deaf ears. I mean, there's been some information that got out somewhere at one point in at least my generation's beliefs that you get the virus and it's better immunity every single time, no matter which virus it is.
Yeah, well, there was the Pfizer trial, for example, included both people who had or hadn't been previously infected. Not Moderna excluded people who've been previously infected, right. But and it was just a handful of people. But if you looked at the people who had been previously infected, there were 8 of them who were then infected after they either got vaccine or placebo. So was infected, got antibodies from being naturally infected. Now we're boosted either with a vaccine or given a placebo. And there were 8 people who then got sick. 7 of them were in the placebo group. 1 of them was in the vaccine group. So it showed you that there was a benefit to vaccination above and beyond the immunity induced by natural infection. So, I mean, that was at least one piece of data. Right. And if you look at these most recent variant trials, where these viruses have to some extent drifted a little bit from the non-variant strain. There was a paper that was just, I saw as a preprint this morning, where they looked at 44 people, sera from 44 people who had survived the infection. Now that they're looking at their convalescent sera, their antibodies induced by natural infection. 22 of those sera did not neutralize this variant strain. The ones that did neutralize it were the ones that were the consequence of a pretty severe infection, where the person had very high levels of titer. So if you had a more severe infection, you're more likely to have a higher level of antibodies. So the critical question here for this variant paper, and I wish they'd done it, was what did they find if they looked at sera from people who'd been inoculated with 2 doses of vaccine? What did they find there? My suspicion is it would be in a much higher rate of people who had the ability to neutralize that virus. So again, another argument for vaccination in addition to natural infection.
And how are you feeling about so I can tell you that on a daily basis, whether I'm doing this for a course or whether I'm doing this because a friend or a family member has asked me about, you know, does anybody have any data on this? You know, I got asked the other day, if I get a negative test, you know, what does that actually mean? Am I not infectious, and so on and so forth. So I'm inundated with this, and oftentimes I am finding that a lot of the information I'm getting is from preprints. How is your thought about the fact that that's just, I mean, I'm not saying that it's a machine and people are just churning out papers, but it sort of has that feel. And I've definitely seen some preprints where, you know, I'm looking at it and saying, well, that's not exactly true, or something like what you said. I really wish they had done this one thing that would have really sewn up their conclusion and given their paper, you know, a much better light as far as the in the scientific community. How are you with this, you know, unbelievable amount of papers that are coming out without peer review?
I know. First of all, I think the statement was something like there are thousands of papers published a day on this, you know, at least online or in some form. Preprint, at least that's something. I mean, I've had to go to Twitter to find, you know, this sort of like from the Bloom lab to find out what their findings were with these variant strains and stuff. I mean, that's where you find information on Twitter. I had a friend who just had to give a hour-long presentation on the variants, and he said, you know, it was all preprints in Twitter. So it's preprints are at least a step above Twitter. This is painful. I mean, when the papers finally get published, you know, we're, you know, miles beyond that. I mean, just in the last couple of days, you know, the phase 1-2 studies for Johnson & Johnson or Moderna or Pfizer just published their phase 3 study. I mean, I was looking at those data as part of the FDA Vaccine Advisory Committee, you know, more than a month ago.
Right. You know, because I'm not a big social media person, but I am on a couple of things here and there, and I'm seeing, you know, people that, how do I say this? The CSOs, CEOs of companies and such, and they're posting New York Times articles, and there's nothing against New York Times. It's great and such, but I mean, it's, you know, there's more likely, in my opinion, based on my reviews and such, there's more likely to be some sort of scientific error in, say, a New York Times article, even versus, say, most of the preprints that are out there. But you're right, it's just anybody is grasping at any information they can possibly get, and like I said, CSOs of companies, I mean, are putting out, like, the New York Times says this and such. And the other thing I thought was interesting is there was a New York Times article that came out recently, and it had a genomic map on it. And I was like, are you serious? And then not only did it have a genomic map, but it had like bubble diagrams of the S protein, of the spike protein. And who is actually reviewing this stuff? And what are they getting out of there? I mean, what do you think about the fact that, and again, I'm not knocking the New York Times, there's nothing against it, it's a great paper, but what do you think about the fact that they're putting up genomic maps and 3D images of spike proteins? I mean, is that something that's approachable for most people?
I don't think so.
Yeah, I don't think so either.
No, I don't think it's approachable, but I don't sort of damn them for doing it. I think that's fine. Maybe it's a small percentage of people that will understand it. I think most people will look at it as they would look at a picture on the wall, you know, just say, that looks pretty.
That's what it must look like. Yeah, exactly. Have you seen anything recently? And I've seen there's a couple preprints, and there's a company that's sort of behind this, about tracking the virus. And I know you're a diarrheal virus person like myself, given all the rotavirus work you did, about tracking potential surges in cases using wastewater. I
haven't seen that. No.
Okay. Yeah. No, yeah. There's some new data. It's put out by a company called BioBot. They're a spinoff of MIT, and they claim to have a way of looking at the virus and having sort of a heads up. If you look and see higher levels of viral RNA in wastewater, that you may end up being able to have a sort of a 4 to 7 day window in predicting when you're going to see a surge in cases, and that's going to be important for local hospitals and such. I just was curious if you had seen that.
I hadn't. It's interesting.
Okay, I can definitely send it to you.
That'd be great.
Yeah. And that sort of brings me to my last question, which is, as far as you know, the variants that are out there, how are you sort of approaching that when you're explaining to people? I mean, are you going to, well, this is how a spike protein binds, and if you've got an amino acid substitution here, the proteins can sort of open up more, and that allows it to bind better, which increases viral titers. Where are you in that sort of area as far as explaining variants?
So I explained that it's an RNA virus. RNA viruses mutate. They all do. I mean, flu mutates, measles mutates. The question is, and it's generally a slow mutator. Coronaviruses are not fast mutators. They're not like flu. But the question is, what's it going to be like? Is it going to be like flu or measles? I mean, flu mutates so much from one year to the next. You need a yearly vaccine. Measles, we've had a measles vaccine since 1963, and the virus has never mutated away. That's the way I generally explain it to the media. Which of the two viruses is this one going to be like? I doubt it's going to be like flu, but I doubt it's going to be like measles too. I think it may be somewhere in between, probably closer to measles, where there will be variants that, to some extent, resist the neutralization by natural infection or immunization. But we'll just have to see. But we really need surveillance. I mean, what do I think we need to do? Absolutely need to do. I was just on a conference call with a group of FDA folks, and we need to have in place clinically that if someone's been naturally infected, known to be naturally infected, or known to have received 2 doses of vaccine, that then gets infected with this virus, SARS-CoV-2, and is hospitalized, we need to identify that virus immediately and see whether it is a variant or whether the person themselves has an inability to make an immune response, but whether this is a variant virus, because we need to get on top of that. That will change your immunization strategy down the line if that's true. I mean, we're doing all those studies in the laboratory, so with these variant strains, UK, South African, Brazilian, I mean, does antibodies induced by natural infection neutralize these strains? Does vaccination neutralize strains? And that we haven't done very much. I mean, it's really the companies that have that sera. It's not a lot of sera. And these neutralization assays are fairly serum intense. So they're still pretty archaic, some of these assays. And I just tried to make a case in the last call that we need a sort of a czar to make sure that we are doing this at a national level and arguably even international level, to take a really close look at these variants, because if a variant arises that resists this vaccine, then you're going to have to go to plan B in terms of your vaccination strategy. And it's hard enough to do this as it is.
Right. And that's actually one issue that I've come about is, unfortunately, there was a lot of comparison by a lot of people that had pretty big megaphone earlier about calling this a flu-like virus. And I've seen so many times, I mean, I've seen on social media recently, I had a friend just pose something about the vaccine. And there was a person that said, well, I have my kids get vaccines, but I don't do flu. And I think the reason why they don't do flu, I think there's probably a couple of reasons. I think one of them is let's face it, I worked on a flu vaccine for a little while. It's hard. It's hard to understand what's coming. There's not many viruses that mutate quite like flu. I mean, HIV, but for the most part, it's a highly it has a lot of different variants and it makes them very quickly. Is something like that just approached with, well, this is not, I mean, how, RNA viruses are not all RNA viruses. Is, there's influenza, which is in a completely different family than this. Again, is that where you land as far as your explanation goes? I'm certainly not going to say, well, coronaviruses are really different in the sense that they're large RNA viruses and they have this somewhat of a proofreading capability. I mean, it's not going to get you anywhere. So where do you just say it's not flu and it's not going to act like flu? So just stop right here.
I don't think it's flu. I think probably closer to measles, but I sort of set up those two paradigms, flu, measles, how dramatically different they are. And we're looking. It's hard to predict this virus. You have to, right. I think anybody who's predicted it, and you can count me among them, have at some point been really wrong.
Oh.
I mean, March 1st or 2nd, when we've had like our first death or two, I had to appear on CNN International with Christiane Amanpour. And I said, because watching what happened in China, which had a population 5 times our size, they had 3,000 deaths, they'd gotten under control. And, I was watching Korea, sort of do all their testing. And I mistakenly thought that what was happening in those countries in any sense was relevant to what was about to happen in our country. And I was just wrong. At the time I said, we had 60,000 deaths from influenza last year. I just can't imagine we would have that many deaths from this virus this year. Now we have like 400,000 deaths. So, if you're going to be wrong, don't just like tell your friend, you're wrong. Tell like an international television audience.
That's better.
Go big, go big.
That's fantastic. I know, and it's funny, you try and talk to, one thing I've noticed is you try and talk to people and, I don't understand, like, how is this virus capable of doing this? And why don't we know we have all these like yourself, we have all these scientists out there. I'm like, it's hard to explain, but large RNA virus doesn't make sense to begin with in a lot of ways. And you try and break it down and say, this is a really, I've just broken it down. This is a really weird virus in a sense. And it's still hard to make that communication and make it solid so that people understand we're not dealing with something that intuitively makes a whole lot of sense and is necessarily going to track data-wise from, like you said, from one country to another. Is that sounds about right?
Yeah, absolutely.
Yeah. Yeah. All right. Well, that's all I have. I didn't want to take up too much of your time because I know you're super busy, but I appreciate very much. I've taken some notes here. I appreciate your time. Anything else before we end our call?
No, thanks for doing this. I think that's great.
Perfect. It was a pleasure meeting you, sir.
Nice meeting you, Shannon.